Once again, I, Ajay Jha, on behalf of ACS India team, welcome you to today's ACS Science Talks session. So just to update you on the upcoming Science Talks, please visit our ACS in India webpage and register for the upcoming session on 13th of August. We have also uploaded the past lecture recordings on the ACS Science Talk virtual library page. Please feel free to visit, like and subscribe and share among the scientific community. ACS, in collaboration with Department of Science and Technology, is hosting the 9th DST and ACS publishing workshop. So follow this link to register for the session on August 11. We'll put the link into the chat box later. To keep you updated with the latest research findings, the product developments, and the events at ACS, we have a monthly newsletter, ACS Insights India. Subscribe today for free. We'll be providing the link into the chat box later. To begin with the scientific session, it's a pleasure and honor to introduce Professor Thomas Hofmann. Professor Hofmann obtained his doctorate and completed his postdoctoral studies in the chemistry department of Technical University of Munich, TUM. He was appointed as professor and director of the food chemistry unit at Münster University in 2002. In 2007, he returned to TUM to serve as the newly established chair of food chemistry and molecular sensory science. Professor Hofmann currently serves in his role as professor and chair at the Center of Life and Food Sciences, Technical University of Munich. He currently serves as the president for research and innovation at TUM since 2019. His today's talk will highlight recent advances in defining our food chemosensory space by means of SENSOMICS approach. He has received several recognitions, including the Advancement in the Application of Agriculture and Food Chemistry Award by ACS, ACS Fellow Award of the American Chemical Society 2008, Firmenich Science Award of the Flavor Industry 2002, Young Scientist Award of the Agriculture and Food Chemistry Division, American Chemical Society 1999, and many more. He currently serves as the editor-in-chief of the Journal of Agriculture and Food Chemistry, ACS Agricultural Science and Technology, and ACS Food Science and Technology. So here we go. So Professor Hofmann, hope you're able to see the slides. Please confirm.
I can see it.
Thank you.
Wonderful. So first of all, thank you for the kind introduction. It's a particular honor to accept this invitation from ACS India to be with you today. And it makes me also proud that many of you dialed in for this presentation today. I would like to introduce you quickly. And first of all, the introduction to the Journal of Agriculture and Food Chemistry. I am the editor-in-chief of this journal now since a bit more than 5 years. On the next slide, you can see that this journal has a long-standing tradition, 68 years of service, so to say. We are internationally set up when it comes to the board of associate editors, 13 associate editors from 9 countries. And the same is true for our advisory board, the EAB, more than 60 members from 25 countries, a global spread of experts from all over the world, which reflects quite a bit also the global authorship we have with many submissions you will see in a minute, which we receive from all over the place. This journal has developed to become a true home of the most significant research in agriculture and food chemistry, and is also a trusted partner for researchers outside of universities like industry, NGOs, and in other fields of expertise. We have a primary focus on high quality original research. Particularly important for us, and also when it comes to submission, is that it deals with innovative applications of chemistry and biochemistry or biological sciences on a molecular level, because we want to increase the molecular understanding of product attributes, of processes, of technologies, of health aspects, and encompass the entire continuum from agricultural practices to food, all the way up to nutrition and health aspects and the underlying mechanisms. So it's a broad scope of the journal that reflects also the broad board of associate editors with a whole lot of different expertise sitting on that board. On the next slide, you can see we currently receive almost 8,200 submissions every year, with a strong increase by more than 40% in the last 5 years. We have accepted currently about 1,500 manuscripts every year. This is plus 37% over the last 5 years, and makes up a strongly increasing impact factor of 5.2 this year. This is a plus of 85% over the last 5 years. So you can see that that journal really is developing well. And what makes me as the editor, but also our office and our associate editors really proud of, is the high number of total citations. With more than 144,000 total citations every year. This is the most cited journal in the field, and definitely one of the most influential ones in agriculture and food science. So on the next slide, you can see that we have different manuscript types. So you could submit original papers on the left-hand side, research articles, which is the major chunk of the journal. We have some review articles, so these are comprehensive reviews. We have perspectives. Perspectives are some kind of mini reviews which deal with a certain element of research and give future perspectives where new research is needed. We have some viewpoints. These are personal viewpoints on certain developments in science. We have also symposia and topical collections. So, for example, if you plan for a symposium and you have a group of authors between, for example, 10 and 30, you could contact us. We are able to build up a special symposium collection to sort of say mirror image the major important content of your symposium. And we have a group of manuscript categories on the right-hand side where you see this broad breadth of the journal. It goes from new analytical methods from the bottom to omics technologies all the way up to agriculture and environmental chemistry. So we have application fields as well as technological and analytical core elements in the portfolio. On the next slide, you can see that we highlight and acknowledge our best papers in two fields every year. One is in the fields of agriculture and agricultural sciences. This is, for example, the winners of 2020. This group of scientists from Italy at that time, they, for example, identified the metabolite of a fungicide to cause a growth disorder in Vitis vinifera, nicely linking plant research, fungicide research with metabolism, and then true impact on the plant growth mechanisms. In the next slide, you can see we have a mirror award in food research. And this is, for example, a work from Japan where they identified a new enzyme from Aspergillus oryzae during the sake process to produce certain ethyl maltosides and ethyl isomaltosides during the brewing process. These two awards come every year and come with $1,000 US dollar award, a nice plaque, and also an invitation to visit one of the ACS national meetings in US, including the invitation to give a keynote lecture on the research. On the next slide, you can see that it's also important to recognize those who do excellent reviews. This is our JAFC Excellence in Review Award, where I can only invite you to participate if you are invited to prepare a review like those guys here, who really do an outstanding job to deliver high quality in-time reviews, which on the other hand is key and important for you as the authors, because also you expect from us that you have a fair and transparent process how your manuscripts are handled. On the next slide, you can see that as we have these 8,200 submissions and we receive many more manuscripts than we are able to publish, we very recently have been growing into a family of journals with the launch of ACS Agricultural Science and Technology on the left side, and the ACS Food Science Technology on the right side in 2021. Both of these sister journals, so to say, if you want, they also publish articles, reviews, viewpoints, and letters. instead of perspectives. With my leadership as editor-in-chief of all 3 journals, I have a deputy editor for each of these new journals. One is Dr. Coralia Osorio-Roa, you see on the right-hand side, for food science and technology. She's a professor at the National University of Colombia in Bogota. And on the left side, you see for agriculture science and technology, we have Dr. Laura McConnell, who is a principal scientist at Bayer Crop Sciences in the United States, 2 really highly ambitious deputy editors who helped to bring these 2 new journals from the ground. So in particular, all 3 journals go through the same quality of peer review process. They differ primarily in the scope, whereas agriculture and food chemistry has a primary focus on molecular work. The other 2 journals have a broader scope, in particular reaching into engineering areas and technology areas in agriculture and food science. So if you submit manuscripts to JAFC that are outside the scope of chemistry and biochemistry in agriculture and food, but still within a broader science, technology, engineering aspect, we will you also transfer to both new daughter journals in order to provide you a quick and easy road to publish your work in most appropriate journals. So if you have any questions regarding the journal, we can exchange on this then during the Q&A session. In the next slide, you see now the title of the presentation, Decoding Chemosensory Systems for Flavor Innovations. So this is a topic we are do research on for more than almost 20 years, actually. In the next slide, you can see why that is so important to understand the chemosensory signatures of our food and agricultural processes and products. You know that with increasing activities regarding disease prevention, food industry tries to reduce fat, sugar, salt, glutamate, add bioactives, make significant changes in current recipes, and that always, in most cases, lead to a severe defect in flavor. And we all know that the sensory perception, the flavor is the key driver in accepting or not accepting a food product. But also when it comes to raw material shortage, you see that new materials come up, other materials are not used to that extent anymore. And that means that it definitely will have an impact on the flavor. And the question is, how can we optimize that by technological means, not by adding artificial flavors? And to do that, in order to drive the processes, you have to understand on a molecular level, what are the key compounds that activate our human odorant receptors and give us the impression of a certain food item. The same is true for alternative technologies like indoor vertical stacked farming, for example, or how we can deliver certain flavors like crustacean flavors without any contaminants of heavy metals, for example, or any meat products without traces of antibiotics. If you want to look for new solutions, we also have an impact on flavor. And that's why to successfully innovate food products, we always need to consider all the edges of this triangle: safety and security, nutritional advantage, and consumer preference. Only if this fits together, we will have really truly innovative food products in the future. On the next slide, you can see a kind of an overview on history. And this is only for a piece of chocolate, for example. We did a literature research on the developments of identifying volatile odorants in chocolate. And you can see it started quite early, in the 60s actually, with the discovery of open tubular gas chromatography. And you can see that then with Moore in 1958, and then with Hofmann and several other scientists, the number of compounds identified in chocolate increased significantly. And then we see always quantum leaps where when new technologies came up, like the first commercially packed GCMS in 1965, you can see a massive increase over the next years when it comes to identifying new odorants. And then the fused silica capillaries did another boost in the 70s. And since then it went up to almost almost 12,000 volatiles identified so far in foods. So a massive plethora of compounds. The question is, which of them do really contribute to the odor? So which of them are key and can be used as, so to say, technological trigger points, which we can then optimize during, for example, agriculture or food processing. In the next slide, you can see the SENSOMICS approach that was briefly mentioned in the introduction. It's in principle a concept that has been developed at TOM in order to identify the key odor and taste compounds. So first, I go through this quickly, and then we will use a concrete example and go individually through the process stages. So first, we have to isolate volatiles and separate them from the non-volatiles. This is number one, by using a special distillation device to isolate volatiles without any artifact formation. Then we have an essentially directed fractionation to identify in number two, where among the different compounds are the really most impactful ones. On those, we then focus the structures of determination, finally followed by quantitation in number four, and rank those compounds based on those activity correlations in order to see which compounds are the ones who have the highest odor impact. Not probably the quantitatively predominant compound, but those with the highest odor impact. That's important. We then on number five, try to re-engineer the flavor because we want to know, are the compounds we identified, are they complete? Do we have identified all the key compounds that are really needed to give in our brain odor impression? If you have this confirmed, then you can go on with human psychophysical studies. You can do chemosensory receptor activation studies, which I will not show today due to time limitations. And then you can use it, for example, on number 8, to go into industrial processes and see how certain process steps do influence our key compounds identified and how we can steer the process conditions in a way that our target compounds are generated in an optimal quantitative balance. In the next slide, you see now an example. Let's start with a glass of red wine. We know that there are many, many red wines out there, and they smell very differently. The question is, what are the key compounds of such red wine? So first, the so-called safe distillation was developed. This is a method that allows you to distill the volatile compounds out of the non-volatile residue at minus degrees under high vacuum. So you do not generate any thermal artifacts. And those are then separated by gas chromatography. You can see that on the right-hand side. This is the trace of the FD response of the detector. And what you can see is that there are many, many minor compounds, but some of them are quantitatively predominant, major peaks as you see in the center. In order now to figure out which of those are odor active, we split the gas chromatographic effluent in a ratio so that you can sniff with the nose the outcome, so the outlet of the gas chromatograph, and can detect those positions in the chromatogram where odor active molecules are present. And these are indicated here by the red arrows. So everywhere they see such a red arrow, there's an odor active compound. All the other compounds are present, but don't smell. They are not recognized by our odorant receptors. On the next slide, you can see how can we, or if you go back one slide, sorry. How can we now focus our structure determination approaches, our structure identification approaches on those odorants that are the most active ones, because they are important. And that's why we need to rank those compounds somehow, the red arrows, in importance. And this is done in the next slide. We developed the aroma extract dilution analysis. That means that the aroma extract that is obtained by the safe distillation is injected into the gas chromatogram. You do the GC sniffing as shown, and then you dilute stepwise, 1-to-1, the aroma extract, and you can see the so-called dilution factors on the y-axis showing that those who have a high dilution factor are the compounds that are in particular very powerful. Give an example, the compound number 6, this is the malty one, for example, you can still smell that compound after more than 8,000 dilution of the original extract. Whereas, for example, the compound 7, the next to this compound number 6, is you can only smell until a dilution of 64, showing that there's a different impact. So we can now focus our structure determination approaches on those odorants who are in particular impactful ones. So the 6, the 17 flowery ones, the seasoning-like, and many others. So if you follow this sensory guided process, then you can get the structures of those compounds. This is on the next slide. So these are the compounds where you see the malty compound is the free methylbutanol. There are two isomers, the free and the 2-methylbutanol. We have a flowery smelling phenylethanol. The seasoning-like compound is the one on the right-hand side. Up there, sotalone, smells like a curry-like smell. And it goes down to the vanillin on the left-hand side down with the vanilla smell. Particular on the right side down, you see the cis oak lactone with a cocoa-like smell. So these are the most important odorants in the red wine. On the next slide, you can see then how we investigate what is the true impact of those compounds when it comes to wine smell. So first of all, you have to quantitate the odorants, because we have to know what is the concentration of those odorants in different red wines. So to achieve this, we quantitate the compounds by means of stable isotope dilution analysis. So that means we synthesize all these odorants in a stable isotope as a stable isotope labeled twin molecule. You can use deuterium labels, you can C label, add the compounds in a known concentration, and then work it up. And then you separate your original analyte, your original odorant in the food from the internal standard that is labeled by means of GC mass spectrometry, as you can see on the upper right. You see the green lines, for example, these are the concentration of the odorant in the food, and the red ones is the corresponding standard that you added. They just differ in the molecular mass by 2 units because we have 2 deuterium atoms added. So when you know the concentration, you want to rank those compounds in the true odor impact. You can do that, for example, by determining the odor threshold concentrations of those compounds. So the question is, what is the odor threshold of the individual compounds that can be smelled by humans? So you see some examples on number 2. Ethanol, for example, a well-known alcoholic compound, has a very high threshold in water. So it's more than 990,000 microgram per kilogram water. So you need a lot of it to be odor active. And this is the reason why in all alcoholic beverages usually have 5 or 10 or 15 or even more % of alcohol, because otherwise you wouldn't smell it. And if you go down, you can see that the 2-methyl-3,4-thiol, this is the molecule at the very bottom. You can see that has a beef bouillon-like smell. It has a 0.00003 microgram per kilogram threshold. So very low, high impact odorant. You need only a tiny bit in order, for example, aromatize a swimming pool, because this compound is so active. And finally, now we combine both. We combine the concentration in the food with the odor threshold concentration. And this is what we call odor impact, or the DOT on the number 3, that stands for dose over threshold. So how many times does the concentration of a certain odorant exceed the threshold, the odor threshold of the individual compound? And if you do that simple calculation, you see in the next slide how the red wine situation is. In the next slide, you see that here, all the odor compounds on the left, you can see the concentrations in the second column. You can see the odor threshold, this stands for OT, next to it, and then the dose over threshold factor, the DOT. And that shows that, for example, acetic acid, which is present in wine in very high concentrations, more than 640,000 microgram, has a dot factor of 115. So it's 115 times above the threshold in wine. That's why you smell that compound. But if you look, for example, the beta-damascenone, which is the least molecule at the very bottom, you can see the concentration is very small, 0.9 only. But the odor threshold with 0.0025 is enormously low. And this is why the dose of a threshold with 360 is high. So it's got, as an odor compound, even more impactful compared to the acetic acid. So that explains why concentration alone does not give you any insight into odor impact. So the dose of a threshold is the key. And you can see, if you look on the right-hand side, odor quality, that none of those molecules smell like red wine. They smell acidic, malty, flowery, stitching, buttery, fruity, cocoa-like, honey, but there's no wine odor. And you will see that later for other examples, that food odor objects like wine or beer or steak or whatever is not generated by individual compounds that's you can see here. On the next slide, you can see then we wanted to confirm that we have, so say, captured all the key compounds. So Richard Feynman, a very well-known Nobel Prize winner in physics, he said, "What I cannot create, I do not understand." And that's why it's so important to reconfirm you're doing. That means all the compounds, 28 odorants now, were combined in natural concentrations in an aqueous solution and compared to the authentic wine. You can see the odor profile, now the aroma profile in blue. This is done by an expert sensory panel. This is the flavor profile of the authentic wine in blue, whereas in red, this is the odor profile of our artificial cocktail of 28 odorants, showing nicely that with this approach, we are able to match the flavor profile out of 28 expert volunteers here of the red wine. So we captured all key aroma compounds. On the next slide, you can see we did that for many foods. We did that for more than 200 foods. And this is a heat map where you can see, so to say, that food odors follow a combinatorial odor code. So you see on the x-axis, you see the 300, more than 200 different foods, and the odor activity value, the important factor which I just explained to you, is color-coded as dots. So if you go to a certain food on the x-axis and you go up to the y-axis, you see the, so to say, the different odorants color-coded. What you can see here is that there are a lot of odorants that sit in the very bottom of this type of chart. You can see on the right-hand side, there is the abundance of the key for odorants. You see that there are many foods that are present, some of them even in 50% of each food. We call those compounds generalists. They are present in many, many different foods, so not specific for one or the other food item. And then there are compounds that are very rare. These are the upper ones. You can see the dots are only present in some of the foods. They're very specific. We call them specialists. And what we also learned is from these more than 10,000 volatiles that have been identified in nature so far, only 230 key odorants are necessary to explain the flavor of all different types of foods. So what we see here in this chart is not more than 230 key odorants, but they are coded in a different way for each different flavors. For one or the other flavor, for a single food, usually we need about 3 to 40 key odorants to explain the flavor of the food. Maybe an alcoholic beverage, maybe tea, vegetable, you name it. This is true for all different types of foods. If you go to the next slide, you see some more examples. Here you see a highlighted section, so to say, of this. You see on the x-axis different thermally treated foods. Like, for example, if you go to the left, cocoa, hazelnuts roasted, deep fried onions, donut, French fries, and rye bread crust at the very right-hand side. And on the x-axis, you see some of the key food odorants. And you can see that, for example, foraniol, which is the upper right, you can see that compound has an odor activity value, so an odor impact in each of those foods. This is a classical generalist. So it's present in each and every of those thermally treated foods. And this is not a surprise, as this foraniol, this is the compound you can see on the upper left, this ring structure, this 5-minute ring structure. This compound is generated from thermal treatment of sugar. And so as long as you have sugar in the food and you thermally process it, you will generate this foraniol. That's why foraniol is a generalist compound. The same is true for example, the second one, which is the methionol, smells like potato. And this is generated from methionine, the amino acid methionine. So each food which has methionine and is thermally treated will present in one or the other way methionol for a generalist compound. On the next slide, you can see now the opposite. You can see, if you go, yeah, that's fine. No, it's fine. Thank you. You can first see a network analysis, which we did. What we did here is to use these order codes that I mentioned and determine the similarity by comparing the order codes. Each food stands now for a dot, for one of those dots, these nodes that you see. And you can see that they are color-coded. We color-coded in green all the foods that are fermented. So you can see in this network on the left-hand side, obviously all the fermented foods have some similarities in order compounds, in order compound profile. Then on the right-hand side in red, we see dry, thermally processed foods, and in the very bottom, we have non-processed raw or aqueous thermally processed foods. So you can see that obviously the combinatorial order codes of foods depend on the history of processing. So what you did with those, what type of processing is used. And this is why food processing is so key and so crucial to determine the right flavor profile. On the next slide, you can see now 3 highlighted examples. In this code, you can see now on the left-hand side, all the alcoholic beverage, beer, wine, whiskey, cognac, etc. So they are very close to each other. On the right-hand side, you see non-processed food, which is, for example, crustaceans, seafood. Also, compared to all the other foods, they are in a certain cloud, so they are different from each other, but they are distinctly different from all the other foods. And in the center, you see cocoa, roasted cocoa or chocolate, which is known to be that some of the compounds are produced by fermentation, whereas others are produced by cocoa roasting. So this is in the middle where all the chocolate products are sitting up there in this green cloud. So if you highlight now the alcoholic beverages, so we just take a deep dive into this small brown network on the left-hand side of the alcoholic beverage. If you do that on the next slide, you see here the code of those compounds, of those products. You see brandy and cognac and Scotch whiskey and American bourbon whiskey, wine, red wine, white wine, Pilsner beer, so all the different type of beverages. Again, color-coded the odor impact. What you see here now, if you click once more, yeah, thank you. You see here, for example, that there's one compound that we call that wine lactone. This is from the chemical structures, the compound at the bottom right. This is the so-called wine lactone. It smells very coconut-like. That compound is exclusively present in wine, in these concentrations, in white wine, in red wine, in different wines, in Riesling, in Chardonnay, in Merlot, in Gewürztraminer, and so on. This compound is formed by a glycoside, which you see in the chart at the upper left. This glycoside is a precursor, which is not odor active, but that is present in grapes. And depending on its concentration in the grape variety, and depending on the craftsmanship of the wine producer, this compound is transformed into the so-called wine lactone. And this is the reason why wines have different levels, different concentrations of that compound. But the other compounds, the other alcoholic beverage, don't have that product. This is a so-called specialist compound. It's distinct because it's a natural product from grapes. On the next slide, you see another example. This is now a compound that comes from oak. So in particular, when you oak mature, so when you ripen the beverage in an oak barrel, then you will always find the compound you see here in the chart upper right, which is the so-called oak lactone. This oak lactone comes from precursors, which we see on the left side. And that compound, again, is a natural product of oak. Is not odor active, but as soon as the oak barrel is heated, because usually you add some heat into it first to charcoal a bit the inside of the wood, then you generate the oak lactone, and that's the reason why you have that compound as an additional odor compound in all oak matured beverages. Also a specialist compound. On the next slide, now you can see the importance of those compounds. What we did here is to compare the odor profile of a barrique barrel matured wine and a steel tank matured wine. So we had the same wine, we split it up in two portions. One was stored in steel tank, one was stored in the oak barrel. We first analyzed those compounds, and you see here in the upper chart the concentrations of those compounds that are different in both wines. You can see that cis whiskey lactone, vanillin, methoxyphenol, 4-ethylphenol, methoxyphenol are significantly higher in the barrique, so the oak matured wine, compared to the steel tank wine. So we did now, in a sense, experiment in order to confirm that those are the ones to determine the typical oaky note in wines. So what we did is first to compare, by using a sensory panel, the barrique wine with the steel tank wine. And there was a significant difference. But if now we took the steel tank wine and spiked that steel tank wine with exactly those 5 red-colored compounds up there in the concentrations explaining the difference between steel and barrique, you could see that none of the expert panel, none of the members was able to differentiate anymore the barrique wine from the steel tank wine that was spiked with the compounds, showing that those 5 compounds, which you see up there, are the key odorants for the oaky smell of red wines that were matured in barrels, in barrique barrels. On the next slide, you can see how does that translate now into odor perception. And there was a very beautiful work done almost 20 years back, actually, by Liang and Jinx. You see that on the left-hand side, a very simple sensory experiment. And what they did was the following. I explain it to you. They made aqueous solutions first of a single odorant. Let me say vanillin. Yeah, you have vanillin solution. And this vanillin solution was presented to sensory panelists, to an untrained panel, a trained panel, and an expert panel. You can see that the expert and trained panelists, 100% of them could identify that, saying this is vanillin. Untrained, about 80%. And now you add a second odorant to this vanillin. Let me say butyric acid. Smells sweaty, for example. What happened? And then you add a third and a fourth and a fifth to the same solution, and always ask, can you distinguish the compounds? Can you identify the odor profiles of the individual compounds? And you see that even the untrained, even the trained and expert panelists were not able to pick up the compounds anymore. Then the number of odorants exceed a number of 4. So that means humans are not able to identify individual compounds in mixtures. And this is important because in nature, you never have single compounds. You always have these mixtures that are presented to you, like wine with the 28 compounds. So what happens now if we mix certain compounds? Does that change the odor object that we perceive? This is what you see on the right-hand side. We had a project some years back where we had a spinach product, dried spinach, so the vegetable. And that spinach had a very bad fishy smell, a fishy off note. And we identified that there are 2 compounds to explain the fishy off note. It was the potato-like smelling methanol and the geranium-like smelling ketone that you see on the right-hand side. And if those 2 compounds that were present in the spinach are present in a ratio of 100 to 1, then you do not perceive it as geranium or potato anymore, but you perceive it as fish. So this was the reason why, although there was no fishy compound in the spinach, this was the reason why the fishy off-note came to the spinach, because due to these two compounds in exactly the right concentration. So it means what is important with this very simple experiment, if you mix those two compounds, is that you need the right compounds in the right concentration. These two compounds, 100 to 1, gives fish smell. And if you now go to the next slide, you can see, next slide. If you do that with other foods, like you have seen with the red wine, for example, down right, you can do the same with king prawns. You can do it with fresh pineapple and wheat beer, always comparing the food item in red with the recombinants or the artificial solution. Pineapple, for example, 10 compounds, wheat beer, 26 compounds, the king prawns, 18 compounds, to explain, so to say, this typical profile of the product. In the next slide, you can see that this reminds us to vision. So how we perceive odor objects is similar to how we recognize vision objects in vision. If you look to that picture here, this is a picture made by Picasso. He made a picture only by a very simple painting, only by 4 lines. So now you have 2 lines. You see 2 lines here, and nobody can tell what it will be. You may think that this is an odor recombinant with 2 compounds. It doesn't give you much. Now, if you click once, if you add the next one, there's a 3rd line to it. So we add a 3rd component to it. Still, we don't get the picture. And if you now do another slide, another line, we can see the picture. And the same is how olfaction works. So with individual odorants, and only if you have the right odorants in the right concentration, so the right length of the lines, if you may call it like that, and the right lines, you get the picture. So also odor object recognition is, so to say, recognized by configural processing, as we call it. On the next slide, you can see which of those compounds are now the ones that really lead us to identify a certain food as strawberry, for example, as pineapple, as mango. So here we compare the odorants of these 3 foods. Mango on the upper side, strawberry left, pineapple right. And we see that some of the compounds are identical. Here's the ones in the center of this Venn diagram. Those compounds are identical, are in all 3, but in different concentrations, but are present in all 3 fruits. Some of them are highly specific to only to one of them. Like if you go to the right side, those 2 compounds with the number 2 and 31 are the ones that are only present in pineapple, but not in the other ones. And the same is true for strawberry. We have 4 compounds that are only present in strawberry, but not in other fruits. So the question is now, which of them are the ones telling you, if you smell something, this is strawberry, or this is pineapple, or this is mango? In the next slide, we did a following simple experiment. What we did was to do, again, artificial cocktails of odorants. And what we did is always to make a cocktail where we have only the shared compounds, which is shared by all 3 fruits, and smell those compounds. For example, the shared strawberry compounds, shared pineapple, shared mango, shared average. And then we asked the sensory panel, what type of fruit do you smell? Strawberry, pineapple, mango, raspberry, although there is no raspberry flavor, but we asked, apple, any other fruit by any other quality. And you can see that with the shared compounds, so with all 3 fruits are sharing, none of the panelists could significantly, this is the p-value that you see color-coded, could significantly detect the odor object. If you now change to the exclusive compounds, so those that are specific for the individual fruits, you can see here in the second box, exclusive strawberry was highly significantly recognized as strawberry. Exclusive pineapple, highly significantly as pineapple, and exclusive mango as mango. And this goes through all the lines. So if in the next slide, you can see, okay, if you go back once, I'm sorry. That means that obviously odor object recognition, so not how is the mango quality or how is the strawberry quality, but how do we recognize a distinct odor object? This is primarily driven through the exclusive compounds, whereas the shared compounds and all the concentrations of the shared compounds only give you differences in odor quality. For example, is the mango a bit more fruity or less fruity, or is more terpenic or less terpene-like? But the object identification, so that you say it's mango, it's not strawberry, or it's not pineapple, that comes by the specific compounds. On the next slide, I want to show quickly at the very end how we can use this to now use it for example, industrial purposes. We do need high throughput quantitation. Up to recent times, the quantitation of odorants in a given food took about 2 to 3 days for one food. Now, with new methods of mass spectrometry, like shown here, for the first time, we succeeded to quantitate even highly volatile compounds, like acetaldehyde, methionol, vanillin, and so on, by means of LC mass spectrometry. If we go on the next slide, we go a bit faster because otherwise we're running out of time. You can see here, we can today quantitate by means of LCMS, that's why we call it united flavor quantitation, odor compounds, like the ones you see on the left-hand side, and taste compounds, like the one you see on the right-hand side, succinic acid, quercetin, which is an astringent compound, anurkic acid, which you can see on the right down, this is a sweetness enhancer, an apple, for example, can be quantitated just in a single run. So it's high throughput united flavor quantitation. In 9 to 10 minutes, you can quantitate those compounds. On the next slide, you can see, we can also use stable isotope dilution analysis as mentioned before. On the next slide, you can see, for example, this is impressive, that in a single run, you can quantitate trace constituents like the beta-damascenone. This is a concentration of 0.01 micromole per liter apple juice, all the way up to the glucose, which is almost 100,000 micromole in a single run. So it's a huge range of concentrations, and that gives us now unique opportunities to high throughput quantitate odor and taste compounds in foods. If you do that, if you go forward, we skip that, and the next one we skip as well. We skip that also. We stay here. If you do that now for wine again, come back to wine. So we quantitated key odorants and key taste compounds by this high throughput methods in different wines, in Merlot, in Cabernets, in Chardonnay, in Riesling. This is interesting. That showed us that all the different wines we investigated always contain the same odorants and same tastants. So there's nothing much specific to it. The only difference that makes these compounds, these wines, different from each other, which makes the Chardonnay specific from Merlot, is the concentration of those key odorants and key tastants. So nature is very simple here. Uses always the same compounds, the same subclass of compounds, and through qualitative differences, we make the differences in flavor. And this is shown by the last slide, where we compare now a total recombinant of the 28 odor compounds in wine and 35 taste compounds we know in wine from two very different wines, a Cabernet Sauvignon, American oak matured on the right side, and the Dornfelder, that's a German red wine on the left-hand side. You can see that odor and taste profiles could completely be matched between artificial recombinant and the original wines by using these same compounds, just differing in the quantitative balance of the concentration. And this is interesting because now we know that we only need to quantitate those compounds. We don't have to do all this discovery work anymore because this code is cracked, so to say. We know what the compounds are. We can go to high throughput quantitation, can, for example, help the wineries to better prepare the next wine for the next year by mixing the blend wines to the right level so that we can always produce in high consistency certain flavors that we want to achieve. So the next slide, and this is my final one, to summarize it, I think what we learned here today is that there's a huge complex world of chemical molecules, but there's only a subset from the more than 10,000 volatiles identified. a subset of 230 odorants that are really needed to explain all different type of food flavors. And among them, the 230 distinct codes, so to say, are used between 3 and about 30 odorants per food item. So it shows this is a truly fascinating decoding of this chemical world, and that gives us now completely new ways to take that into industrial applications, or even to deliver like an odor piano, where it could play different songs, so to say, by using a keyboard of 230 odorants. So thank you for listening.